Always a great pleasure to be attending the AP, even more being part of a talk in the Animal Physiology Commission. I am the presenter of this article here, and from the 6 authors of this paper, 4 of us are attending the conference here in Tallinn this year. And this conference, this section is particularly special for me, because in 2008 I was attending the World Congress on Animal Production in Cape Town in Africa, and you had the very same section there. So it's very nice to see that section come back to the surface here in Tallinn. So my presentation will go through a typical scientific outline. Some of the results are still somehow preliminary, but I think that they're quite interesting. That's why I decided to share that with you. So there are 3 main pillars in my talk. One is feed efficiency, second one is immune system, and the third one is metabolomics. And it's all that surrounded this situation here, when junior comes out, and there's a lot of physiological events happen at that moment. So we start with feed efficiency. This is a quick slide just to show you how important is feed efficiency in the beef cattle production. And these are all data from my research program. So despite of all the numbers that I have in this slide, the main points here are, when you look to the 10% most efficient cattle in a herd, you might be saving 600 kilos of hay a year per animal in feed consumption. And then if we convert that into dollar value, you're talking about savings of $140 a year per animal, just for the sake of being more efficient. And because females in the beef herd, they can last for a long time, they can be there for 10 years. It's quite different from the poor dairy cows. And in over 10 years, that animal, just for the sake of being more efficient, can be saving $1,400, which means that you're pretty much, the animal is buying the 50% of his own replacement just for being superior. So our biggest challenge here is to identify such animals. The second component, it is the immune system, and I'm taking 2 key approaches here. One is when you look to the immune system in relation to the immune response. So you see those pie charts that when rats are subject to an immune challenge, that changes quite a bit how they have their protein accretion patterns. A good example is what happened here in the liver. You can see that in the sick animal, we have a lot of protein metabolism or accretion to the liver, and in the healthy animal, that's a smaller chunk. And myself work with beef cattle. That's quite important, because what really matters for us is tissue accretion. So that's the relevance of that. And the second aspect of the immune system has to do with the background, the basal energy requirements that are related to the immune system. The literature is not so clear in identify the breakdown of the basal energy requirements in terms of immune system, but you know that the immune system takes a big chunk of that proportion of the basal energy metabolism of the animal. And the third one, it is metabolomics, and here I'm taking more like a comprehensive view about it, because there's a lot of focus on the metabolomics, but we can never forget about the big picture. And you know that through the entire chain, changes on different aggregation levels probably have an effect on the metabolic profile. So the metabolomics can be an interesting way of studying the animal physiology. And one example that I have here is a contribution that we gave at the AP in 2015, where we look at the metabolomic profile. That's just one of the metabolites, alanine, and looking at heifers over the circadian period, at the open stage or puberty, and then early gestation, late gestation, and then you could see a distinction of the alanine profile in that context. So with that, there is certainly a variation in the efficiency of feed utilization. And then we are here checking if there are any use of looking at the immune system and at the metabolome to potentially find some biomarkers that could be making easier the identification of animals that have superior feed efficiency. Therefore, our objectives in this study were to quantify the humoral immune response or parameters of the immune system, and also look at the blood and the colostrum metabolome in beef heifers, and from 2 perspectives. one from the feed efficiency perspective, another one from the physiological state around the calving time. So for this study, we had access to 36 pregnant heifers. We monitor individual feed intake of those heifers for over 500 days. So went all the way from weaning until they're dropping their first calf. We did measurements of body weight every 2 weeks. We did ultrasound on a monthly basis. So the ultrasound traits are important for us to infer about leanness and also fatness. So that's why we did those 4 measurements there. We also knew the day gestation of those animals, their age, breed composition, and a few more things. So for doing feed efficiency, we use as our feed efficiency traits residual feed intake. And we're a little bit old-fashioned here. You're doing a feed intake assessment with Cullen Gates. And then our prediction of feed intake, so our model for those heifers, include the actual body weight of the animal, the body weight variation, and then measurements of ultrasound, and also include the age of the animal and the age gestation, which are so important sources of variation for those animals. And with that information, we could calculate our residual feed intake, and just a reminder that a negative RFI, it is a more efficient animal. So in terms of the immune system, we decided to challenge our heifers with a novel antigen. So we vaccinate them against the ovalbumin. And we did that with a prime dose 33 and then 26 days prior to calving. And also we collect quite a bit of blood samples. So we did blood sampling at the baseline, booster, calving, and nursing. And nursing here means 47 days post-calving. Also, when the cow give birth, we also collect their colostrum. And with that, we process our samples using ELISA, and we measure total IgA, total IgG, and also the specific IgG to ova, so the specific antibody against the ovalbumin. And how we dealt with this data, we classify our animals according to efficient and deficient, so we did a categorical analysis here, which was also used for the metabolomics. And also we had a categorical analysis according to the physiological state, so baseline, booster, nurse, and calving. We did a very simple stats on there and compared the square means among those classes. When it comes to metabolomics, I use this equipment here that I have access in the province that I'm from in Canada. Did an NMR analysis, and we did our data analysis in a few different fashions. The first one was a principal component analysis. So basically, it's more like a black box approach when you look at the whole metabolome of the animal, and then you create your principal components, and then we could be running these components in the context of feed efficiency and physiological state. And moving to next level of this, we did also look at the chemical shifts. So this was a bit more laborious. Went through the metabolome and then identified the peaks according to literature references. And then with those peaks in each animal, we could run Pearson correlations between the different metabolites and feed efficiency measure at different stages, as outlined before. So this is all for methods, and then let's look at some results. Since I am not an immunologist, it's always good to take a look in their data and see if it makes sense for like a very basic principle. And that's exactly what we're doing here in this slide. So on top line, you have the concentration of the different immunoglobulins in the colostrum, and then the bar graphs, you have the concentrations in the serum. And thankfully, things are making sense. Both the total IgG and total IgG ova are about 10 times higher in the colostrum than what you have in the bloodstream of the animal. And also when you look at the IgA, we have a very small concentration in the colostrum and a way higher concentration in the bloodstream. So there is quite a few references showing that should work around that, so we are good in this regard. And being on a safe ground, then we can look at our relationships with feed efficiency, and that's what you have in here. So the top line shows the IgG ova for efficient and inefficient animal. And this data is showing us that the efficient animal had a higher level of specific ova. And then when you look to IgGA, the inefficient animal had a higher level of IgA. So there aren't much results from this type of data for us to compare in the literature. So that's why I pick one paper from our lab that's currently in review. This paper also done in heifers in a larger population, and also looking at ova specific. There wasn't difference for the ova specific in this group. However, when we looked at the IgM secondary response or the booster, we could observe that animals that are more efficient also had a higher concentration of that specific immunoglobulin in the bloodstream. And then moving towards the metabolomics, this first graph here shows the physiological states. So you could see that the metabolomic profile has some distinction according to the four metabolic states that we look at. However, when we look to feed efficiency, the data is quite overlapping. You couldn't distinguish only on PCA a clear path between efficient and inefficient animals. So moving to our next level here, when we look at the chemical shipments, there are some comments here. There's still work on this. But firstly, we see there's the UA all over the place. So those are unsigned beings, means that they're still working towards identifying those metabolites. We don't quite sure who they are at the moment. We could see the lipids are present in a few situations here. And I think that's really worth to dig further in lipids. And I'm saying this because recently we got this publication here when we measured cholesterol over the circadian period in beef steers, and you could observe that efficient animals consistently have a lower cholesterol level, and that's exactly what we're looking for a biomarker, like quite consistent over time. And back to this, we also see some amino acids here that still need to work a little more, and the same is true for glucose. I just want you to pay attention that glucose here in the buster situation had a negative correlation, and then at calving we had a positive correlation with feed efficiency, and that's quite interesting. From my experience, I have been seeing that the best way to assess if animal is efficient or not, it is to disrupting the homeostasis, and this is a clear situation of disrupting the homeostasis here, so it's something for us to further consider. So where we are with this? This is just a small sample of this study. We have a lot more going on here in terms of look at them with different physiological states, also organs, cellular biology, and metabolomics. So certainly have a lot more to show and share with you in Croatia next year. And just for a quick conclusions here, that both were successful in establishing different physiological states according to the immune and metabolomics. It seems that there's promising results in the immune system that could be used for develop future biomarkers. And I still think that there's a few steps behind for getting that situation when it comes to metabolomics. We need to study a lot more of that. And with this, I'd like just to thank this funding agencies that support this study. And if you're interested in this type of research that they're conducting, we have our full profile available on ResearchGate, and also quite a few videos at our channel on YouTube. Thank you.